So in this next image, what we're seeing is some of the data that Mendel collected. He crossbred hundreds of thousands of peas, and he was able to actually find some interesting patterns with their dominant and recessive traits. And so here are some of the ratios of what he saw in terms of the phenotype. If you notice, most of these you can actually see that if you estimate it, you get a 3-to-1 ratio of the dominant to recessive phenotype being shown. And so Mendel spent a lot of time looking at these characters of the plants, or what we know now as traits of the plants. And he was able to see, again, that there was 2 contrasting forms of each trait, which we know as an allele. What he saw was that in the F1 generation, okay, that first cross, was that, or actually what he questioned himself is that, is it always the dominant purple phenotype? And so this is something that he really looked at over and over and over again. And he did see that in most cases he had a certain number, like 705 purple flowers to 224 white flowers. But what you want to do is take a little time and look at this data that he collected. It is going to be important for us to see the observed data and this expected data. And so just think about, well, what formula have we used already where we've seen observed data and expected data? And later on you should be able to use this data to determine one of those tests. And the test I'm specifically talking about is the chi-squared goodness of fit test. Remember that this statistical test allows you to see if your observed data is similar to the expected data, and if the difference is significant or not. Okay? So what did Mendel not know, even though he was able to figure out a lot of these traits, and that, hey, the purple flower was dominant over the white flower? What did he not know? And so Mendel didn't know about DNA yet. He didn't know that DNA was inside of a cell. And he didn't know that there were genes or DNA that was divided into sections. And unlike us, he also didn't know that these coded for amino acids in a specific order that eventually formed a protein, which eventually formed your trait. So we're going back to this idea of DNA being that blueprint of life, DNA needing to go through some processes like transcription and translation to produce a protein or trait. He didn't know any of these, but he was able to figure out and determine a lot of data for us that we wouldn't have known otherwise. So then we have a couple more terms that Mendel didn't know and that we do know more about now. So you have your chromosomes. Your chromosomes were just coiled up DNA. We know that they're inherited through meiosis. And yesterday we also mentioned the term alleles. And alleles are just going to be different forms of a DNA sequence. And I'm sorry, this is getting so sloppy. Here you have a picture of homologous chromosomes. And here you may have the same gene or information for the same trait, but you may have different versions of that trait, different alleles. So here our A's would represent the alleles, the bar you see represents the gene or trait, and the two purple chromosomes represent your homologous pair from possibly mom or dad, or vice versa. So a little more important, and probably something that you don't know as well, is going to be Mendel's 3 principles. And so he got these 3 principles by breeding some purebred or homozygous green plants, green peas, with purebred or homozygous yellow peas with green seeds. And notice that you have actually in this example, this is going to be a dihybrid because we have 2 traits that we're looking at here. This is a dihybrid. 2 traits. You have your pods and your seeds representing your 2 traits. So the first principle that Mendel came up with is the principle of dominance. Your dominant version is always going to be a capital letter. The allele is going to be represented by a capital letter. Version is going to hide your recessive, and this is always going to be your lowercase letter. The dominant you can think of as the stronger version. And really what the allele is showing or representing is a DNA sequence. So as an example, we could have a DNA sequence that says AA, T, T, C, C, G, and this could represent our dominant allele, big A. But then we can have the same gene, but a different allele, let's say T, 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 C, C, G, and this could represent our recessive allele. All that the letters are representing is a section of DNA or a gene, different versions of that gene. So don't forget that. Don't get caught up in the capital A's and the lowercase a's. Remember that all of this goes back to the DNA sequence. Even though the dominant version can hide your recessive allele, you can still pass on the trait or the recessive trait. And a lot of times an organism can have or have the hidden trait, and this would be called your carrier. Then our second rule is the principle of segregation. And the principle of segregation you can also think of as separation. Basically means that during meiosis, your homologous pairs divide and reduce to the haploid state in the daughter cells. So each parent donates half of their chromosomes. If you want to think of this as here's 2 alleles, here you have mom, here you have dad, you have the same gene on there. This is a set of homologous chromosomes. The third principle is going to be the principle of independent assortment. And in the principle of independent assortment, this just means that one trait doesn't determine we'll add in the word inheritance of another trait. These traits are going to be independently inherited. So basically, that means different genes are on different chromosomes. So different genes are on different chromosomes. These three rules only work for traits that are simply dominant or recessive and do not work for other traits. So we know, for example, some sex-linked traits are not independently assorted, and they are related to each other. For the principle of segregation, overall, this is true unless you have non-disjunction and have a syndrome like Down syndrome. And principle of dominance doesn't always work, especially when you think about blood types or codominant traits like sickle cell anemia. Those are also not going to follow those rules. But we'll just stop there for today. Please take a minute to look over these. I know they're a little sloppy. And answer the rest of the questions on your lecture tools.